So our third speaker today is Dr. Gary Wu from, excuse me, the University of Pennsylvania. And Gary's going to be talking to us about the role of diets, standardization, and characterization. Clearly, from the preceding talks of the last day and a half, diet is of critical importance. So, Gary, welcome.
Thank you very much. First of all, I want to thank the organizers for inviting me to give this talk. I hope I don't disappoint, because as I went through to think about what I would actually talk about on this topic, I realized there's actually vanishing small information about this. So what I'm going to do is talk in the first half of my talk about the value of studying diet in animal models, a lot of which you've already sort of gotten a sampling of, so that will make some of those slides go by very quickly. But then what I thought I would do is talk about something that hasn't really been brought up too much so far, and that is the translatability, because one of the bulleted items, I think, for this symposium was to think about how these preclinical models are actually relevant to human biology. That is an area that we try to work in. I'm the mouse wet bench researcher that does stuff in cell culture also, but I do work within a group that tries to translate into human biology. So I will focus on that particular topic in the second half of my talk. So challenges to characterizing the effect of diet on the human gut microbiome. Here are all the challenges. Humans are poorly adherent to dietary regimens. I'll show you some slides about omnivores and vegans. We love working with vegans because they will be compliant with their diet. We hate working with omnivores because we can't trust omnivores in terms of their diets. So the current tools to characterize dietary composition and intake are imperfect. There's a reciprocal nature of dietary composition to maintain isocaloric consumption. And so it's difficult to identify what is actually responsible for the observed outcome. If you change one thing, you have to change something else. Diet can have profound impacts on a host biology independent of the gut microbiota. And then both intensive controlled feeding experiments and large outpatient cohort studies are expensive and challenging to complete in humans. So that's the value of studying animal models and the interaction between the diet and the microbiota. So your tight control of defined diets over long periods of time. Multiple biological replicates are feasible. Germ-free animals can be used to examine the effect of diet independent of the gut microbiome, and defined microbial consortia or complete human gut microbiota studies can be performed in notobotic animals, and cause and effect relationships involving diet and the gut microbiota can be determined. I'm going to show you towards the end of my talk that this is both a strength and a weakness when we think about translatability into human domain. So these are the topics I'm going to cover. Don't get scared. It's going to go by very, very quickly, okay? So functional impact of diet on the murine gut microbiota and its metabolome. All right, so it's fundamentally important. You already know this, that diet not only shapes the composition of the microbiota, but serves as a substrate for the microbiota to produce a lot of small molecules that then, after first pass metabolism, circulate widely throughout the body. And that's the notion for why the gut microbiota has an effect at organs distant from the mucosal surface. So what do I mean by this? Well, diet effect on composition of the gut microbiota's impact on disease has been demonstrated by Jane Chang. And so the consumption of a particular milk fat causes the mouse to deliver more sulfated bile acids into the gut microbiota, and you get a bloom of sulfate-reducing bacteria, Bolophila wadsworthia, accelerates more Th1 immune response, and exacerbates colitis in an IL-10 knockout mouse. Effect of diet on metabolite production by the gut microbiota, impact of disease. There are many examples of this. This has already been discussed, and this is the notion that what you eat in terms of fat delivers choline to the microbiota. It converts choline into a gas that we absorb. It's oxidized in the liver by flavine monooxidases. TMAO is a biomarker for cardiovascular risk, and Stan Hazen has gone on to show that this accelerates coronary vascular disease. So you already heard about this yesterday. I'm a gastroenterologist, so we got in the field many years ago because we were interested in inflammatory bowel disease. So these are diseases that affect probably close to now 2 million people in the United States. And these are devastating diseases. They are chronic in nature. They require almost lifelong immunosuppression associated with various types of extraintestinal manifestations. Now, we know that there's a very strong environmental effect on development of inflammatory bowel disease because the incidence of IBD is rapidly rising worldwide. This is very similar to many other immunologic diseases that we see on the rise associated with industrialization. This began several decades ago in North America and Europe, and then Japan followed, and now in China and India is seeing a very, very rapid rise of inflammatory bowel disease. As some of you may know, biologics, some of the primary drugs we use for the treatment of inflammatory bowel disease, are not readily available in China. So this is of enormous concern to them in terms of their healthcare policies as well as the suffering of these individuals. So this is a very strong environmental effect. Now, when we think about diet, there actually is a dietary therapy for the treatment of inflammatory bowel disease, and these are defined formula diets. Some of these are elemental diets. This is used as first-line therapy in Europe, in Japan, in Israel, a couple places in Canada and United States. And the reason it's not more widely used, and this is specific for Crohn's disease, one of the two extremes of the inflammatory bowel disease spectrum, the reason it's not used more widely is that these are actually very difficult to take. They are monotonous. Some of them are unpalatable, and they have to be delivered by a nasogastric tube. Now, despite their efficacy, we really don't understand how they actually work. So we, as other groups, are very interested in this paradigm on this slide here. So I didn't have time to show you all the data, but believe me that their diet is epidemiologically associated with inflammatory bowel disease. There's unquestionable information really supporting the notion that a gut microbiota is a significant driver in the development of IBD. And diet, we've already heard, can influence the gut microbiota and its metabolites. So our approach has been to start in the human domain, start with therapeutics that we already know are effective, and understand the proof of concept. Actually, what is the mechanism by which why they work? Do they work independent of the gut microbiota? Do they work through the gut microbiota to impact upon disease? And the idea is that if we really understand the biological principles, then we could define better diets for patients with inflammatory bowel disease. That's the approach that we're currently taking. And we're trying to systematically deconvolute a very, very complex issue, largely actually in humans. Now, it's important to know these defined formula diets in the treatment of Crohn's disease are most effective when consumed completely in place of a normal diet. That's why we call it exclusive enteronutrition. If you start to eat whole food, the disease comes back. And so this, just to let you know how little we understand about this, there are two fundamental questions that we need to address. And that does exclusive enteronutrition provide something good for patients with IBD that's not abundant in a regular diet? Or does the consumption of exclusive enteronutrition exclude something that is bad for patients in IBD that has been introduced into our regular diet? So we are beginning to see this in animal models, that our diets are fundamentally different than they were several decades ago. And now there are things that are now being introduced into our diets that, at least in animal models, seem to alter the microbiota and favor inflammation. Now, again, I'm not saying that artificial sweeteners and dietary emulsifiers cause inflammatory bowel disease or any type of disease. These are largely mouse models. But it is a very interesting notion, supports the idea that our diets are fundamentally different, and that could be one of the several environmental factors that are playing a role in increasing incidence of certain types of diseases like inflammatory bowel disease. Okay, diet and defined bacterial consortia in notobotic mice. So I'm going to show a couple of slides on Jeff Gordon because he's really sort of led the field in some of this work. So this is a study using studying monozygotic twin pairs as well as notobotic mice harboring a 15-species model human gut microbiota, and asking what is the impact of fermented milk product, essentially yogurt, on the composition and the function of the microbiota. And in both humans and mice, there was really no significant change in the composition of the microbiota, but there was a transcriptomic signature that was seen both in mice and in humans. You can see sort of the patterns mimic each other. It's very specific, tended to be more with plant polysaccharide metabolism. So it's a notion that you can use define notobotic mice to sort of mimic and understand the biology in humans in response to a particular dietary intervention. And this would be fermented milk products. And Jay has actually done some really nice studies and trying to sort of deconvolute the complexity of diet on interacting with the gut microbiota using the defined consortium of 10 organisms. But now we're beginning to see the next level of these types of analyses, where computational biologists are now beginning to deeply into various types of clinical metadata, including the microbiome, to make predictive responses. In this case, glycemic responses, and in this particular case, looking at diet in general. How does the human actually respond to diet using some concepts that we understand about the composition of the gut microbiome? This is not just the organisms, but their genomes. In this particular paper, they actually use genomic metabolic modeling, basically genomic flux modeling, to make predictions about how certain microbes would respond to environmental stress of diet and what type of products that they would make. And they came up with a model that would predict, and I think primarily amino acids in plasma. So this is, I think, a very tractable system where we're beginning to see more and more in the literature. Okay, diet and humanized microbiota in mouse models. Jeff talked a lot about this yesterday. So, you know, the whole point is that you can transfer the microbiota into nootrobotic animals and then look at the effect of diet. And it's a really, really wonderful model system and story there. Impact of the gut microbiota on mammalian host metabolome. So, you know, one of the challenges, as I mentioned, is so how much of the effect of diet is specifically on the host is independent of the microbiota. And one of the values of animal models is you have germ-free mice, so you have defined colonies that are in mice, and you can ask how much of it is host, directly effect on a host, and how much of it is through the microbiota. And so there are studies looking at the plasma metabolome of germ-free versus colonized mice or rats treated with antibiotics. There are hundreds of plasma metabolites that are actually different. Now in humans, we actually don't know this. We actually have a study called FARM where we're trying to map this in humans. But we probably know maybe a dozen or two small molecules that are definitively from the gut microbiota that are in human plasma because we can't make those molecules. And the idea is, can we think of a strategy to actually map this in humans? But this is the value of studying, for example, a germ-free mouse versus a colonized mouse, because I personally believe that a lot of the input, at least in the metabolome, in terms of diet, is independent of the gut microbiota. And I'll show you a slide or two about that. All right, so the last part of my talk is predictability and translatability of preclinical studies to humans. This is really what I want to emphasize the most. So Eldon's actually showed this. I'm going to present this again in sort of a different way. And this is the extreme and consistent effect of diet on the murine gut microbiota. This is just a paper that we published many years ago, but this is something that you see very consistent in the literature. It's a very, very strong effect of diet on the mouse microbiota. So in this particular study, we had two mice. We had a wild type and had a knockout mouse. I won't even tell you what the knockout is. It doesn't really make any difference because you can see that on a high-fat diet, there was an extreme alteration in the composition of microbiota dominated by firmicutes and some proteobacteria. This has actually been demonstrated previously. And it was independent of whether or not you were wild type or knockout. So on this principal coordinate analysis, again, you've sort of seen these types of analyses before. Each dot is a different community. And dots that are closer together are more similar to each other. Communities that are further apart or more distinct are actually further apart in terms of dots. So along principal component number 1, it's 28% variance. So 20% variance in the composition of the microbiota is actually determined by diet, because the high-fat diet's over here and the normal child diet's over here. This is extreme variance. The genetic impact is only about 6%. I mean, it looks big here, but it's really not because the axes are actually different. So the genetic effect is relatively modest compared to the variance imparted by diet. Now, they do actually interact, as Eldon's mentioned, because the knockout mice moves from here to here, and the wild-type mouse moves to here. So there's an interaction between genetics and diet, but diet leads to very significant, massive changes at the phylum level that are consistent between mice, independent of genotype. All right, the translatability into humans. So we did this study. It was actually quite simple-minded. It was essentially driven by our notion that people who live in Africa have a very different microbiota than people living in the United States. So we thought, well, it must be diet, because people who live in Africa have a very different diet. So we thought, well, if we just took individuals that eat largely plant-based diet in the United States, what would their composition of microbiota be? What would be their metabolome? So as I mentioned, we've been studying vegans and omnivores for a while. This is a cross-sectional study. We had 15 vegans, 16 omnivores. We collected a lot of dietary information, samples from metabolomics and gut microbiota. So that's what I'm going to show you in like 2 slides here. This is previously published. I'm going to just show you like 3 slides to make a particular point. So we know that the diet between a vegan and omnivore is very, very different. In principal coordinate analysis, omnivores are over here, vegans are over here. They're very different. All the micronutrients are down here. They're classified in certain categories. And largely, vegans are here, omnivores are here. Higher consumption of certain types of micronutrients denoted in red are lower in vegans and vice versa. Okay, so we know that. Diets between a vegan and omnivore are very different. But then the plasma metabolome between a vegan and omnivore are also very different. So we did this with Metabolome. 363 small molecules are right along the bottom here, can be classified in certain categories up here. Vegans are here, omnivores are here. By and large, plasma levels of certain metabolites that are higher in vegans are actually lower in omnivores and vice versa. This is really, really extreme. And so, when we sent around these plasma samples to Metabolome, one of the computational biologists actually called me back and said, what are these samples? I've never seen, this must be a disease versus health type of thing. I said, no, these are healthy individuals. And they said at that time, this was one of the largest separations in the metabolome in the human plasma they've actually ever seen in healthy human subjects. It's so extreme that, in fact, if you do a random forest analysis, you can predict with 94% accuracy whether or not you're vegan and omnivore simply by looking at 30 small molecules. As you would predict, the urinary metabolome is actually very different from the plasma metabolome, all right? So both diet and the urinary plasma metabolome dramatically different between omnivores and vegans. All right, so what about the composition of microbiota? So this was actually a surprise to us. So in contrast to the metabolome and the diet, the composition of the microbiota, now this is again 16S tag sequencing, similar to what we did in mice, similar to what was done initially in Africa and the United States, looking at different types of communities. You can see that now the dots in terms of color intermingle with each other. So the composition of the microbiota between a vegan and omnivore is actually not that different. Statistically it is. It's at a p.04 level, but it's a very modest difference. And even diversity is only minimally greater in the vegan than in an omnivore. But I just showed you data showing that, again, the plasma metabolome and the diet are extremely different. So this was actually unexpected. That was not our hypothesis. But then we went back and we thought about a study that we had previously published a couple of years ago where we did a controlled feeding experiment. We called it CAFE. So there were 10 healthy volunteers. We locked them up in the hospital for 10 days, and we fed them 2 different diets, a high-fiber diet, low-fiber diet. One was reciprocally, so the low-fiber diet is high in fat, and high-fiber was low in fat. 10 days, we collected stool samples every day, and the results are summarized here. So each individual is a different color. Each ball is a different day of the study or a different stool sample. So you can see that for each color, there's always one ball that's different from all the other balls of the same color. That ball that's always different is day 1 of the study. So that means that within 24 hours of consuming a different diet, your microbiota actually does change, but there are a couple of very important points here. So you can see how different everybody is from each other. This is this intersubject variability that we see in human populations. It's one of the largest source of variance in human studies. The impact of diet, although highly statistically significant, is very modest relative to intersubject variability. The second point is that even though half the individuals on one diet and half the individuals on another diet, the way that you move is almost stochastic. You can't actually predict which direction you're going to move. And that, I have this notion that where you move because of an environmental stress of a different diet, it sort of depends on what you actually started with. And the other thing is that these groups didn't actually converge over time. So people on the same diet, at least within 10 days, would not converge. They wouldn't become more similar to each other. So when we published this a couple of years ago, we thought, well, that must be because we didn't have these individuals on diets long enough. Long-term diet might have an effect. And you'll see in the literature that long-term diet will have a bigger effect than short-term diet. And I do believe there's probably some element of accuracy about that statement. Nevertheless, I do point out that our vegans in our study were vegans for greater than 6 months, yet they're not that much different from an omnivore. Now, you will see actually in the literature reports, and this is work by Peter Turnbaugh, and there's like 1 or 2 others about rapidly changing, reproducibly altering the human gut microbiome in terms of diet. Peter's a fabulous investigator. I really do like this study, but we do have to keep in mind that the structure of the study. In this particular dietary intervention, this is a ketogenic diet. This is devoid of any carbohydrates. And there's another study by Steve O'Keefe where he took Africans and put them on African Americans, put them on an African diet. An African diet is 65 grams of fiber. Just to give you perspective, vegans, where you think eating a lot of fiber, that's about 30 to 40 grams of fiber a day. So there's no question that really, really extreme diets will lead to more consistent changes in the microbiota. But I've talked to both investigators. Intersubject variability still was dominant even in those studies, even in very extreme diets. So I want to just give you a perspective about the impact of diet on the microbiota. So the bidirectionality of the gut microbiome investigation. So in the animal world, I shamelessly ripped this off from your website here. Sorry about that. So you have a defined environment, conditions, defined genetics, monotonous diet. And for that reason, we do this for a reason, because we want to have a high signal to noise ratio. We want to be able to see something so we can prove proof of concept, cause and effect relationships with a modest size cohort. I mean, that's what we're all talking about here. But in humans, we're free living in a highly variable environment, genetic diversity, variable diet, and we have very low signal to noise ratio. So it is absolutely a challenge. But thinking optimistically about this, small effects over large populations can be very highly impactful. So I think some of the things we see in the microbiome are likely in human populations due to the variance that we see with all these other factors may have a relatively modest effect size, but that doesn't mean that it's not significant. Because let me just use the example of this FDA drug development. If you have a drug and there's a very, very slight signal that maybe there could be an adverse event, that's why they do post-market surveillance over millions of people over time, and you can have an effect on many, many, many people. Think about the whole idea about food additives, just as an example. The number of people with a relatively small effect size over large populations over time could have an enormous effect. All right, so challenges. When I think about it, coprophagia in terms of diet, that's a huge issue. I don't know how you address that. Response of endogenous gut microbiota to diet differs in magnitude and consistency relative to humans. I already demonstrated that. Mouse chow in studies is monotonous relative to variability in human diet. We talked a little bit about this. Murine digestive physiology in response to diet is different. Bile acids are different. Fermentation, as you know, mice are fermentative organisms. They have a large cecum. Humans don't have a large cecum. They're not fermentative. So short-chain fatty acids are likely to have a bigger effect on certain parameters in mice than they are to humans. The impact of DNA in mouse chow. So when we treat with antibiotics, we've done what Jay has done, try to extinguish the microbiota using antibiotics. When we sequence, we see lactococcus. So lactococcus was from casein. It's actually in the food. So this can be a source of variance in your sequencing results. Lack of mouse chow standardization as a possible cause of variance. The composition, closed formula, open source diet, batch-to-batch variations, phytoestrogens, all these things are probably going to have a significant effect on the composition of the microbiota. Whether or not you use a sterilized versus non-sterilized diet, we have evidence, and I didn't have time to show you, that a defined consortium will be less resilient to environmental stress if you use a non-sterilized diet. Intuitively obvious, but we have evidence for that. And then feeding behavior, ad lib versus fasting. And then animal welfare issues, you know, you have a limited ability to exclude the transfer of the microbiota via coprophagia between housed mice. You know, you can't really put mice for any period of time in wire bottom cages. And then I do know that there are some investigators that are singly housing mice. But then, as I understand it, mice are very social animals. So it's likely to change their phenotypes. And undoubtedly, that's going to be a stress on the mice. Impact of long-term dietary interventions, extreme modifications, caloric intake, impact on the mouses could be very significant. And even things that we consider to be grass, generally regarded as safe, and food supplements at high concentrations obviously could have adverse outcomes in mice. So thank you very much. I really appreciate it. Oh, I forgot the most important slide here. So this is our group, all right? So Rick Bushman, Jim Lewis, and I sort of started the human intestinal microbiome group. We have a lot of investigators. We all work together. Some of that translational work that we do really requires teamwork. So thanks.